Hi, my name is Julian Enrique Sanchez Guajardo, and I'm going to present my argumentative oral exam about beta-glucan extraction from SRBCI. First of all, the first question was, the number of unit operation can be decreased. Which unit operation will you remove or replace? First, we need to know about the characteristic of the designed bioprocess that we present. The general operation is very simple. In this case, is the simplest the process that we recover from literature with the objective that to make it accessible for the brewery. As you can see in the figure 1, the general operation consists only in a pretreatment, solulysis, alkali acid extraction, and finally a dried. Of course, let's take in account that if general operation would have one or more unit operations with it. But is the simplest protocol that we can see. And by that reason, we can decrease the number of unit operations. But the second question is, which unit operation will you remove or replace? If we see the result obtained in our purification table, we can see the yields of mass, carbohydrate, and beta-glucans. And in the three cases, the operation that present the worst yields is the bed meal or the disruption of the cells. So a sustainable solution for the replacement of a unit operation, it can be used another mechanical method that it has been reported that are the most efficient at small and large scales. It's sustainable because you will be avoiding the use of chemicals that can be harmful for the environment. But if personally, instead of remove or replace a unit operation, I will optimize the bed meal because it had been reported that the most efficient and reliable method to disrupt the cell wall of the yeast is the bed meal. So you can change the parameters of the bed meal, and this can be another solution to change the unit operation and having better yields. The second question was, what are some of the challenges associated with the scaling up of the beta-glucan extraction process from the lab pilot scale, and how can this be overcome? The first one that I see is the mass balance, that because you will need to dimension the unit operation to satisfy the demand from the project, considering the operational volumes and the equipment and the fluxes that we'll be working with. How you can overcome this part? Knowing the fluxes in the lab scale process. How measuring the volumes that you are working in each unit operation and the mass. With this knowledge, you can make a mass balance, and with this mass balance, you can scale it to an pilot scale. The second one is the scaling parameters. That you will need different parameters because the parameters that you work in a lab equipment will not be finally the same ones that the pilot scale. If you want the same result, of course, because the proportions are different, the volumes are different, the operation is different. And how do you overcome this? You need to use correlations or mathematical models to make that the result that you obtain in the lab equipment will be the same to the pilot scale. One example is the one that I show in the figure 4 from Maybury et al. And was used in our project. That it was the mathematical model for scaling from a basket centrifuge to a disc centrifuge. That the basket one is not continuous, it's a batch, and the disk is a continuous one. So it was complicated to make this mathematical model, and it was reported in the literature. The third question is, how can the purity and the quality of beta-glucan extract from expanded brewer's yeast be assessed? And what are some of the key analytical techniques that can be used? For this, to analyze the purity and quality of beta-glucans, there is some analytical techniques. There's others analytical techniques that can be used for analyzing the process. But in this case, For only the purity and quality of beta-glucans, one is the HPLC, that is a liquid chromatography, that will help you to obtain the beta-glucan content, purity, and yields. There's an example in the figure 5 of the HPLC that we obtained from our project for beta-glucan. And one analytical technique that is not specific for beta-glucan, but can be used for the purity, is the total solids. That is the difference of weight after evaporation of water from the samples. Other options of analytical technique that can be used is the use of kits. One example is the enzymatic kit of Megazyme, mentioned in the presentation, that what the name said is an analytical technique made of enzymes. And how do you read this? In this case, this kit is with absorbance, and will let you know the beta glucan context. And the last analytical technique that we present, from the difference that there is in the in the that can be used finally, is the spectroscopy infrared, of course, that it will give you the structure of the beta glucan, because it analyzes the molecule, the complete molecule, and can give you information about the quality of the beta glucans of your that you have extract. And that can be all. Here is the reference, and as you can see, there is different points of view about the beta glucan extraction from SRBSI. You can see it from a scaling part, you can see it like the units operation, and you can see it as the analysis of your results. And I think that's all. Thank you.